Hello guys, good morning. How are you all? Okay, we'll wait for one more minute for others to join. Audible this case, please check the pool options. Okay, good morning. Today we'll start the class without wasting any time. Anyway, it's already 10 o'clock, right? In today's class, we are revising the biomolecules. So you maybe have a question like, why we came to the biomolecule? Because already we are at 12th levels, right? So just to remind you guys, in this particular crash course, I designed the curriculum in such a way that, okay, so along studying the 12 important topics, we will also include the important some 11 topics, majorly from this cell cycle and the biomolecule chapter, because from these three chapters, you can expect more than 5 or 6 questions. Okay, sometimes the question number went up to like 9 questions from these particular chapters. Okay, so these chapters are very important. This will also be basics for the other chapters also. That's why I just wanted to have a quick revision or our revision of the complete biomolecule. Okay. So we'll see if anyone is not joined or any friend of yours need to be joined, inform them and tell them to join quickly. Okay, so we'll start the class. So as usual, before starting the class, we have a question here. The question is super easy. Just read the things. The two functional group characteristics of sugars are, they are asking, which functional groups will be present in the sugars or fats? The first one is hydroxyl and methyl. Second is carbonyl and methyl. Option C is carbonyl and phosphate. Option D is carbonyl and hydroxyl. Okay, so you have 30 seconds of time. This question asked in me 2018. So answer this simple kind of question. I'm giving you 30 seconds of time. Okay. Okay. I only got one correct answer, all the others given the wrong answer. Just guys, remember, is there any methyl group in the sugar? Think about glucose. Okay, glucose contain a methyl group. No, there is no methyl group. You can remove the option A and option D. Okay, does sugar contain any phosphate? I'm not talking about nucleotide. I think about like glucose. Does it contain any phosphate? No, right? There is no phosphate in that. So you can remove the option C also. What is the correct answer? The correct answer is carbonyl and hydroxyl group. Okay. So which groups will be present in the sugar? The groups which are present in the sugar are the carbonyl and hydroxyl. Okay. Carbonyl and hydroxyl group. Got it? Any doubt in that? If you have any doubt, let me know. Okay, so carbonyl and the hydroxyl group. We'll go for the second question. The major role of minor element inside living organism to act as. Then asking what is the major role of minor elements? Okay, so option A, cofactor of enzymes. Option B, building blocks of important amino acids. Option C, constituents of hormones. Option D, binder of cell structure. For the people who wanted to know, okay, the minor elements mean something like molybdenum, manganese, magnesium. Okay, so those elements. So these are like minor elements in our body, right? So what's the function or what's the major role of this particular element, do you think? Okay. Let's see. Again, that is again time I'm giving. Let's see who will give the correct answer. Great. Okay, yeah, the time is over. This question is asked in 2003. Is it for the binding of cell structure? No. Is it a constituent of hormones? Majorly, but it's not a major role. Okay. Binding blocks of important amino acid? No, that's also not the correct answer. Then why? What is the important reason or important role of this minor element? These act as a cofactor of the enzymes. We'll study what is cofactor, what is the coenzymes in the end of this particular chapter. For now, we just have to remember that the minor elements which is present in the body, or sorry, minor element which is present in our body act as a cofactor of the enzymes. Remember, this is given in the textbook. You may think that this may be out of syllabus question. No, it's given in the textbook, clearly written, okay, in the similar kind of the statements. Okay, if you are not sure about it, go and read the biomolecule. Okay, so we'll come to the actually things. Okay, in today's topic, we'll normally cover carbohydrate, proteins, lipids, and enzymes. First thing we'll start to discuss is about a simple thing about biomolecule. Okay, when we talk about biomolecule means any organic molecule which have an importance in the living system or which involved in the formation of the living system. Okay, these biomolecules are divided into two types again. One are micromolecule, and another one is the macromolecule. This micro and the macro is totally dependent upon what's the size. Okay, if the size is very less, then it's included in the micromolecule, something like amino acid or monosaccharides, those kind of things. Macromolecule are the one the size are comparatively big. That means any polysaccharides or any proteins or any lipids. Okay, those are involved in the macromolecule. Okay, the first thing we learn is metabolites. Metabolites means any molecule which gives the energy, okay, or any molecule which involved in the metabolism process, right? And those are called metabolites. Metabolites again divide to primary and secondary. Primary metabolites are the one which help in the growth, okay, which help or involved in the growth. That means these give the energy. This give the energy, and using this energy we will be growing. Okay. Example, I will give carbohydrate, protein, fat, right? All these are the primary metabolites. That means what's happening? They are giving the energy, right? They are giving the energy. What are secondary metabolites? These are not helping the growth. Then why they are present in our body? These are present in our body to give the defense. Okay, these are involved in the defense. Okay, what is defense? If you consider any living system, right, this is normally invaded by the different types of the parasites and the pathogens, right? See, these particular metabolites help to protect the body from these particular pathogens and parasites. We have some examples here. Metabolites, again, we divide into different categories, okay? Some of them are pigments, metabolites. Example, as you know, carotenoids or anthocyanins, chromin, all those kind of things. Next, maybe alkaloids. Alkaloids will be like morphine or the codeine. Okay, morphine. Have you ever taken anacin? Anacin contain morphine. Okay. Next, terpenoids. These are monoterpenes, diterpenes. Essential oils. If you take lemon, it have an essence. There's a lemongrass oil. Or toxins. Some of them are highly toxic. Think about like from the seeds of the resinous communis, we extract a chemical called ricin, okay, or abrin, or lectin. Lectins are a group of compounds with multiple rings. Example, conformation A, or some of them we take as a drugs, especially like cancer drugs like vinblastine, or an antibacterial one. Example is curcumin. Okay, and some are polymeric substances. That means they form a long chain. Example, rubber, gums, and cellulose. Okay, these molecules help in the defense in the plants because only plants, fungi, and bacteria normally produce it. Usually plants. Okay, we utilize them for our personal purposes, right? For the humans, we extract them and we use it for our purpose. Example. All these are the example, right? Think about like tea. Why teas have a bitter taste? Because they have a secondary metabolite. And why coffee is related to our body? Because it have a secondary metabolite called caffeine, right? Caffeine, for the plant, it produced it to avoid the pathogen, right? For us, we are drinking to energize ourselves, right? So that is how the things work. Understood? Then we'll go for primary metabolite one by one. First one I want to go is the carbohydrate. Okay, what are carbohydrates? Okay, carbohydrates are the hydrates of the carbon in a simple term. Okay, these are the hydrates of the carbon. they normally have a ratio of like a carbon, hydrogen, and oxygen in 1-to-1 ratio. So, like in a nutshell, I can write it something like CnH2nOn as a general formula. Okay, take an example of the glucose for your reference. Okay, glucose has 6 carbon, 2 N, that means 12 hydrogen, and again 6 oxygen, right? So this is general formula for the carbohydrates. Okay, and carbohydrates also normally referred as saccharides. Okay, normally referred as the saccharide. Majorly, this particular word came from the word saccharide, that means sugar. Okay, because the first identified carbohydrate is the sugar, so what we are using from ancient time. From there, this term called saccharide came. Okay, these saccharides or the carbohydrate can be divided into 3 major types. One is the monosaccharide. Second one is the oligosaccharide. Third one is the polysaccharide. Can you give me any example for this? Any example or anything like that? Any example? Remember, okay, when we talk about monosaccharide, disaccharide, oligosaccharide or the polysaccharide, so these are the different components, like how many kinds of monomers it contains. Okay, monosaccharide means, as you can guess, it contains a single unit of the sugar. Okay, single unit of the sugar. Based on how many carbon molecule is there, this monosaccharides again divided into something like trios, that means 3 carbon, or tetros, okay, or it may have it having 4 carbon, pentos, which is having the 5 carbon, or 6 hexos, 6 carbon. Okay, so it's divided to based on how many carbon is present. It's again divided in like hexos. Next, all the other things will come. Okay, but as you think about the chemical group, the saccharides can be divided into majorly 2 types. Okay, those major 2 types will be aldoses and ketoses. Okay, what are aldoses and ketoses? I think you already guessed. The monosaccharide which have a aldehyde functional group called aldoses. Okay, let me take an example, something like glucose or galactose. Okay, or mannose, all those examples. If that particular monosaccharide have ketone as the functional group, then it's called ketosis. Example I can take as the fructose. Okay, remember the fructose? Fructose is a fruit sugar which is the highest sugar level naturally available. Okay, naturally available, having highest sweetness level is the fructose. That's why like fruits taste sweet, rich in the sweetness because they have the fructose. Okay, so these are the monosaccharide. When monosaccharides combine to form, when 2 monosaccharides joined, then they will form the disaccharide. If 3 monosaccharides join, then it's called the trisaccharide. Okay, so that is how oligosaccharides are, in a simple term, I can say it like 2 to 9 units of sugar. 2 to 9 units of sugar. I will take an example, something like disaccharide, trisaccharide, tetrasaccharide, okay, trisaccharide, tetrasaccharide, so on, okay, until 9, okay. So majorly we'll study about disaccharide. Okay, example of the disaccharides is, I will take major one, sucrose, maltose, and lactose. Maltose and lactose. Okay, these are made up of different monosaccharide units. Like lactose is made up of glucose plus galactose. Okay, maltose is made up of glucose plus glucose. Sucrose is made up of glucose plus fructose. Okay, so different monosaccharide unit combine to form different disaccharides. Okay, different monosaccharide units combine to form different disaccharides. Okay, so there is one more thing I wanted to hear. Okay, these monosaccharides are normally called as reducing sugars. Okay, why it's called reducing sugars? Okay, so these monosaccharides, since they have an open aldehyde or ketone group, they will reduce the Cu2 plus ion or cuprous ion, which is present in the Fehling reagent or Tollens reagent. Okay, I think you're already familiar with the Tollens or the Fehling reagent, right? You studied in the chemistry. So what happens? All the monosaccharides normally have aldehyde or ketone open group. These open groups normally reduce the carbon ion which is present in the Fehling or Tollens reagent. That's why it's called as the reducing sugar. If I consider the disaccharides, okay, disaccharides also normally show the reducing property except sucrose. Sucrose is a non-reducing disaccharide. Other than that, most of them also show the reducing property. But as we go frontwards, other polysaccharide, those are non-reducing because they do not have the open groups. Okay. So that's about the oligosaccharides. Then we will directly jump into the polysaccharide. Polysaccharide means more than 9 units of sugar. More than 9 units of sugar. Okay. So these polysaccharides are like many number of sugars. Okay. It's again divided to two types. Okay. One is the homopolysaccharide. Okay. Homopolysaccharide. And another one is the heteropolysaccharide. Heteropolysaccharide. What is the meaning of this? Homopolysaccharide means it is made up of a single type of unit. Okay, single type of unit. Think about like glucose. Only glucose joined continuously formed. That is a homopolysaccharide. Okay, think about it's made up of the different types of the units, something like glucose, fructose, galactose, mannose, everything. Then it's called as the heteropolysaccharide. That means different type of monomers. Okay, different types of the monomers. So those are the major things we can mention. Okay. Homopolysaccharide example, you can take out the cellulose or the starch or glycogen or the chitin. Okay. Heteropolysaccharides are regularly comparatively rare, glycolic acid, those kind of things can be considered as heteropolysaccharide, but that's not important. Okay. Then we'll directly jump into the protein. Okay. What are proteins then? Proteins are the polymer of amino acid. Proteins are polymer of amino acid. AA means amino acid. Okay. These particular proteins, when I consider, is majorly composed of 20 amino acids. This is majorly composed of 20 amino acids. Okay, so proteins have a general, amino acids have a general structure. I can write it something like this. Okay, here is a 1 carbon and here is our R group. That means which can be changed. Okay, H, another side is CO minus and another side is the NH3 plus. Okay, because this carbon is attached by 4 different group of atoms or 4 different groups. This carbon, I think you guys already know about this, you studied in the chemistry, right? So amino acids have chiral carbon, okay? Amino acids have the chiral carbon except glycine. Glycine is that will be later, okay? So as you can see the structure, it have a 2 functional group. One is the carboxylic acid functional group, right? Carboxylic acid functional group, and another side it have okay, the amino acid end which contain carboxylic acid group is called the C terminal. Okay, the amino acid end which contain amine group is called as the N terminal based on like what's present. Okay, it's called as the N terminal. Understood? So as you can see here, this particular amino acid can be present in 3 different forms. Okay, one form, what happens? The CO minus will be there and amine will be there. That means it's showing a negative property. And another time, what happens? Amine group showing a positive and COH will be there. It's a positive, but it can be present in an intermediate stage. Okay, that means having both of the ions, like negative and also the positive. This kind of property of the amino acid is called as a deuterionic form. Deuterion means what? Deuterion means having both positive and negative ends. Both positive and negative ends. Positive and negative end is called as the Zwitter ionic form. Okay, Zwitter ionic form. Those are the basics for the amino acid or the protein. Okay, detailed about amino acid, I think you guys studied in the biomolecular chemistry and also in the molecular biology. Right. So we'll move on to the next one. So before that, we'll study about some of the important protein factors and what they will do in the cell. Okay. So one of the important protein in our body is the collagen. Okay. It acts as an intercellular ground substance. Remember, it is the most abundant protein in animal kingdom. Okay, in animal kingdom, because it's not present in the plants. This is the most abundant protein in the animal kingdom. There's the collagen fiber. Next is the trypsin. You can say there are like enzymes also formed of the proteins, right? And there are a lot of hormones also made up of the protein. Example will take as the insulin. Yesterday only we studied, right? And the antibodies, which is secreted in your body to fight against infectious disease, those are also made up of the protein that you guys studied in human health and disease. Okay, and receptors which help for the access into the cell or access to the smell, taste, hormones, everything again made up of the protein. Some of the transporter molecule, example, GLUT4, glucose transporter. GLUT4 means glucose transporter 4 molecule. Okay, this kind of transporter molecule which is present in the plasma membrane also made up of the protein, right? So protein is very important. That's why we call protein as the building blocks of the body. Building blocks of the body. Building blocks of the body, right? Building blocks of the body, we call it. Understood? Okay, we'll go for the next one. Amino acid and protein conformation. Okay, when we think about amino acids, so amino acids are combined to form proteins. How it happens? If I take an example here, okay, so as you can see here one carbon is there, and think about like here we have amine group, okay, NH2, and think about here is another carbon which has this COH group. So this is belong to COH, and here NH2, R2, R1, HH. What happens? Water will be removed from here. Okay, water will be removed from here, and it forms a bond, something like this. Okay, so this kind of a bond is called as a peptide bond. This particular bond is called as the peptide bond. So amino acid forms peptide bond with other amino acids and form the proteins. I can write something like this, right? So this particular chain is called as the polypeptide. Okay, polypeptide. If number of amino acid increases and if it have a function, then it's called as the protein. Okay, then it's called as a protein. Some of the amino acid which is given in the textbook, I also mentioned here. You just have to remember the structure of these three, especially in the case of biology. That's alanine, glycine, alanine, and serine. How it is different? If it is simple, okay, the simplest amino acid is the glycine. Okay, this is the simplest because here in the R group, a H is there, one hydrogen is there. So because it's a simple and this is the only amino acid which will have non-chiral carbon, right? It's not a chiral, right? Because it have a 2 H groups, right? So you have to remember the glycine. If you replace this H in the R group, if you put the methyl group or the CH3, then it will become alanine. Okay. Instead of CH3, if you put CH2OH, then it will become serine. You have to remember these three structures. Okay. So when the protein formation happens, when there what happens, protein will form different types of the structure. Based on their types or based on the complexity, we divide proteins into 4 different conformations. Okay, first one is the primary structure. Primary structure. This is the simplest form of protein or simplest form of the polypeptide. Here what happens? Amino acid will form a chain. Amino acid form a linear chain. Okay, so what happens here? Okay, it's a very basics. So here the sequence of amino acids form a chain. Okay, primary structure means there is no particular shape, it forms a chain. Okay, second one is called again secondary structure. Okay, secondary structure. In the secondary structure happens when the primary structure are too long. Here, a new steric hindrance or the new steric relationship between the amino acids develops, which forms two different types of the structures. One is the alpha helix and another one is the beta sheet. Okay. Here what happens is in the alpha helix, these particular amino acids form a helix structure, a helical structure like this. In beta sheet, it will form a sheet-like structure like this. Okay, so this is how it forms. As you have to remember here, okay, what are the bonds are implementing? If I consider the primary structure, only peptide bond are present, okay. But when you come to the secondary structure in the alpha helix and beta sheet, along with the peptide bonds, you can also see disulfide bridges because of the cysteine, right? And also hydrogen bonds in between them. And also you can see van der Waals forces, okay, van der Waals force. Right. So it can form two types of the secondary structure. One is the alpha helix and another one is the beta sheet based on how it's called or how it's arranged. You have to remember that in the primary structure or in the secondary structure also there is only one polypeptide is there. As you can see here, it's not made up of the several polypeptide. It is made up of the single polypeptides. Okay. So let me show you the alpha helix. As you can see here, this is one ultra structure of a protein, right? As you can see here, the red color coiled around things are there, right? These are the alpha helix, okay? These are the alpha helical structures, okay? Beta sheets cannot be seen in this structure, so that I will show some other day, okay? So we'll go to the third one. If the more complexity happens, then it's called as the tertiary structure, okay? Then it's called a tertiary structure. What is tertiary structure? If these secondary structures form a three-dimensional structure, okay, if these particular structures, secondary structures form a three-dimensional structure, then it's called as the tertiary structure. Tertiary structure is bending and folding of the secondary structure only, okay. It will be stabilized again with the ionic bonds, van der Waals forces, disulfide bonds, hydrophobic bonds, and the group interaction, functional group interactions. Okay. The fourth or the highest form is the quaternary structure. Highest is the quaternary structure. Quaternary structure, what happens? It happens in a simple term, I can say it is like it's a three-dimensional structure in multiple protein. Okay, with multiple protein. Okay, remember primary, secondary, tertiary structure, it because of a single polypeptide, whatever happens is related to single polypeptide. Okay, but quaternary structure means whatever the tertiary structure, if it happens with more than one polypeptide, then it's called as the quaternary structure. Okay, let us take an example of the hemoglobin only. Okay, hemoglobin is made up of the four different types of protein. One is two alpha chains and two beta chains. As you can see here, blue is the alpha, okay, two alpha and two beta, okay, they combine to form a single protein, right? Multiple proteins are multiple polypeptides are combined to form a single protein. So this is called as the quaternary structure. Understood? In a simple form, how can I say? If a single polypeptide form a linear chain, it's called a primary structure. If the single polypeptide form a structure forming the disulfide bond and hydrogen bond on van der Waals force, forming either helix or a sheet, it's called secondary structure. If this secondary structure fold three-dimensionally, then it's called a tertiary structure. If more than one polypeptide combined to form a protein, then it's called as the quaternary structure. Okay, quaternary structure. Understood? So these are the major thing about the protein. You guys have to remember one more thing, that the proteins are very sensitive. Okay, they can only function in certain particular temperature and particular pH. Okay, above or below that, they will get disintegrated. Okay. So we'll move on to the next one called lipids. Okay, what are lipids? These are group of substances with hydrophobic nature. Okay, what a group of substances with a hydrophobic nature. Okay, hydrophobic nature. Majorly, we know, every one of us know about the fat, right? So we majorly know about the fats. So we can simply call it as like polyesters, right? Poly alcoholic esters. Okay, we can say polyalcoholic, long chain polyalcoholic esters. Okay, so if I will directly go for the what's the classification here, lipids are divided into three major types. One is the simple lipids. Second one is the compound or the conjugated lipids, compound or conjugated lipids. Third one is the derived lipid. Okay, these are not lipids, but these are derived from the lipids. Okay, derived lipids. Okay, so first we'll go for the simple lipids. Okay, simple lipids are like no, true fats actually. These are the true fats. These are again maybe either fats or oils or waxes. Okay, or how can I divide? Simple is two types. One is the true fats, another one is the waxes. Okay, true fats contains oils and it also contains different types of the fats. Okay, different types of the fats. Okay, so you can consider all your oils and everything in here. Okay, oil, butter, ghee, everything in here. Simple. Okay, waxes are, these are esters of fatty acid with a long chain of monohydric alcohols. Waxes you can see in the plants, in the chitins, or in our body. Those are belongs to simple kind of the lipids. Conjugated lipids means these are combined with the other types of the biomolecule. Let me take an example. Something like phospholipids. Phospholipids. What are phospholipids? Phospholipids are the one where lipids are combined with the phosphate group. Or glycolipids. What are glycolipids? Glycolipids are the one which is combined with the glucose, sorry, which combined with the saccharides or the carbohydrate. Okay, or maybe we can take also example of lipoproteins. Okay, here what happens, lipids are combined with the protein. These are called conjugated. That means they combine with the other biomolecule. Okay, third one is derived lipids. Okay, derived lipids means these are not true lipids. These are derived from the lipids. Something like steroid, sterol, hydrocarbons, or I will take an example of vitamins, something like vitamin A, vitamin D, vitamin K, etc. Okay, these are not lipids, but these compounds are extracted or derived from the different lipids. Okay, different lipids. Okay, major term here in a simple lipids we divide are so saturated and unsaturated lipid. That's the very important term here. Remember, this is in the simple lipids. Okay, simple oils or the fats are divided into saturated and unsaturated based on what kind of bond they are presenting. I think you can see the structure here, saturated and unsaturated. Saturated fatty acids are the one which have a single carbon-carbon bond. They will only have carbon-carbon single bond throughout their chain. Don't consider the functional group, okay? But in a chain of the carbon, main chain, it only have a single single bond, carbon-carbon single bond will be there. Because of this carbon-carbon single bond, what happens? It will stack easily on another. Okay, it will stack easily. So in the room temperature, these saturated fatty acids will form a solid structure. In a room temperature, they form a solid structure. What's the example? I will take an example of the butter. Right, I can take an example of the butter. Right. But in unsaturated fatty acid, what happens? So there is a double bond will be present in between. Okay. Because of the double bond, what happens? The angle will change. The angle will change. Because the angle is changed, what happens? This cannot stack. It cannot stack one on another easily. It cannot stack properly. So because of that, what happens? An unsaturated fatty acid, normally liquid in the room temperature. Example, you can take oil, right? Oils are the example. What happens? Unsaturated fatty acids are normally present in a liquid form. Okay, if you can guess, so saturated, the food is stored in the saturated form in the animals. Okay, in the animals, it's stored as the saturated form. Unsaturated fatty acid, you can find it in the where in the plants. Let me take an example. Think about butter or the ghee. From where it's coming from? It's coming from the animal, right? It's coming from the cow, or it may be coming from the some other type of the animal, like a goat or the sheep or camel, right? So what about unsaturated? Think about palm oil, right? It's unsaturated, it's in liquid form, right? Or you can take an example sunflower oil, right? Or you can also take an example of what all other kinds of plant-based oil, olive oils, right? Everything is liquid because these are unsaturated oils. Okay. These unsaturated oils are again classified into how many double bonds are present. Okay, if it contains a single double bond, it's called mono unsaturated. If it's contained two double bonds, it's called di-unsaturated. If it is contain more double bonds, then it's called polyunsaturated, or in a simple term, it's called PUFA. Okay, polyunsaturated fatty acid, or PUFA. Okay, PUFA are good for your diet. If you have any oil can in your this one, what's that, in your home, go and check that. There it will be mentioned it as contains PUFA. Okay, what's the PUFA? PUFA means unsaturated fatty bonds, that makes it comparatively better for our diet than the saturated tests. Okay, so these are some of the examples which is given in the textbook. The structure of fatty acids are simplest one, and the phospholipids, lecithin and triglyceride, and also the structure of cholesterol. You have to remember these things because one time they given the structure of cholesterol and they asked which molecule it is. Okay, so those are the basics. We'll move on to nucleic acid then. What acids are the polymer of nucleotide? These are the polymer of nucleotide. These are the polynucleotides. This can be simply divided into major types on RNA and DNA. We'll go through the difference later. Okay, nucleic acids are composed of three major components. What are the components here? The components are, first one is sugar, and the second one is the nitrogenous base, and third one is the phosphate group, right? Phosphate group. These are the major three components. Sugars are again divided into two types of sugars will be present. These sugars are the pentose sugars which are present in the nucleic acid. The first one will be the ribose sugar, which have a regular structure. Okay, that means something like this. And the third one is the deoxyribose sugar. Deoxyribose sugar. What happens in deoxyribose sugar? In deoxyribose sugar, one oxygen missing in the second carbon position. In the second carbon position, one oxygen is missing. That's what's called as the deoxyribose, right? And nitrogenous bases again divided into two types. One is the purine, and another one is the pyrimidine. Pyrimidine, right? Purines are again two types. One is the adenine. Okay, and the second one is the guanine. Okay, pyrimidines are again divided into types. Okay, one is the thymidine, cytosine, and uracil. Uracil. There is no classification in the phosphate group. Okay, so let me tell you one more thing here. Okay, when sugar combined with a nitrogenous base without the phosphate, this is called as the nucleoside. Nucleoside. When sugar combined with nitrogenous base plus phosphate group, then it's called a nucleotide. Then it's called as the glycosidic bond, N-glycosidic bond, N-glycosidic bond. And the fifth carbon atom of the sugar is joined with the phosphate group with ester bond, with the phosphoester bond. Okay, phosphoester bond. Okay, understood. So ribose and RNA. The measure between DNA and RNA is like what type of the sugar it contains. DNA contains deoxyribose sugar and RNA contains ribose sugar, right? What is the second difference? DNA contains 80 nitrogenous bases, that means adenine, guanine, thymidine, and cytosine. RNA contain AUGC, adenine, uracil, guanosine, and cytosine. Okay, RNA do not RNA instead of thymidine, they have uracil as base. This is the very basics about the nucleic acid. Okay. When question comes, try to answer the question. Question is simple. Which of the following have carbohydrate as prosthetic group? They are asking which particular group have a carbohydrate prosthetic group. Option A, glycoprotein. Option B, chromoprotein. Option C, lipoprotein. Option D, nucleoprotein. I am launching the poll. Okay, you can answer within the 30 seconds. Okay, try to answer within the 30 seconds. I'm just waiting. Or you can also answer in the chat box. Okay, or you can also answer in the chat box. Which of the following have carbohydrate as the prosthetic group? Glycoprotein, chromoprotein, lipoprotein, nucleoprotein. Okay, still last 10 seconds. Last 10 seconds. Okay. So I'm ending the poll here. Okay. Only 60% given the correct answer. Okay, only 60% given the correct answer. We'll see what's the correct answer. Okay, so they are asking about carbohydrate as the prosthetic group. All of them are protein. That means we have to check what's the other thing. Nucleoprotein is the correct answer or not? No, it's not a carbohydrate. Remove this. Chromoprotein. Chromo means color. That means this is also not the correct answer. Lipoprotein means it contains lipids. So this is also not the correct answer. Then what's the correct answer? The correct answer is glycoprotein. Right, glyco means not only glycogen, glycoprotein means any carbohydrate containing protein. Okay, understood? Then we'll directly dip into the enzymes. Okay, enzyme part, what is enzymes and all the different terms. Okay, the term enzyme is given or coined by W. Kuhne. Okay, W. Kuhne. He is the one who given the term enzymes. Okay, you just have to remember that most enzymes I'll write it in this way. Okay, I will write it in this way. All enzymes are protein. All enzymes are protein. But all protein are not enzymes. Understood? So not enzymes. All enzymes are proteins only, but all proteins are not enzymes. So here one exception I will write, except ribozyme. Okay, that you will study later. Okay, so what we are saying is like except ribozyme, that means some RNA molecule act as an enzyme, except those molecules. All the other enzymes are protein, but all the proteins are not enzymes. Okay, enzymes are divided into two types based on the structure. Those will be the like simple enzymes, and the second one is the conjugated enzymes. Okay, conjugated enzymes, or the conjugated enzymes, or it's also called as the holo enzymes. Okay, holo enzymes. What are simple enzymes? Simple enzymes means these particular types of the enzymes have only protein part. These enzymes contain only protein part, nothing else. I will take an example here. Example, you can think about this like amylase, trypsin, pepsin. Okay, all these are the example for the symbol. That means they have a only protein part. This acts as an enzyme, only protein part, nothing else. Okay, but when you come to this particular conjugated or holo enzymes, okay, So here we come across with the second thing. Here the parts are divided into two types. One is the apoenzyme. First one is the apoenzyme. Apoenzyme means protein part of the enzyme. Protein part of the enzyme. Protein part of the enzyme. And the second one is the non-protein part of the enzyme. Non-protein part of the enzyme. This non-protein part of the enzyme again divided into two types. Okay. First one is the our coenzyme and second one is the cofactor. Coenzyme. Second one is the cofactor. Okay, so let me explain what is this. If you think about the enzyme contains a protein part, right? In the conjugated enzyme, the protein part is called apoenzyme. As you can see here, this protein part is called apoenzyme. Okay, and the non-protein part, as you can see here, right? These are also important for the enzyme to function. These are called as the non-protein part. It's again divided into coenzyme and also cofactor. Okay, coenzyme and the cofactor. Okay, what is the difference between coenzyme and cofactor then? Okay, so if a particular part attached to the apoenzyme, apoenzyme loosely, okay, or loosely attached part is called coenzyme. Okay, firmly attached part is called cofactor, or it's also called a prosthetic group. It's also called as the prosthetic group. Simple, understood? If any part which is loosely attached to the apoenzyme is called coenzyme, if any part which is firmly attached to the apoenzyme is called as the cofactor. Okay, so if I take an example, the cofactor example is FAD. Okay, flavin adenine dinucleotide. I think you studied in the respiration in plants chapter. Okay, but I can also take example for the coenzyme is the NAD. Okay, nicotinamide adenine dinucleotide, NAD plus. So this also you studied in the respiration in plants chapter, right? So these are the major types or the classification of the structure of the enzymes. Okay, so sometimes you just also remember along with NAD, certain metal ions also act as a coenzymes. I think you already know about it, right? Manganese or molybdenum, okay, or also the magnesium, these are act like a coenzymes. Okay, these metal ions act like the coenzymes. These are called as the metalloenzymes. Okay, if metal ion is joined to an enzyme, then it's called as the what it's called as? It's called as the metalloenzymes. It's called as the metalloenzymes. Metalloenzymes. Okay, so we move on to the next part. Enzymes are classified into 6 major groups. Remember, this is very important. Okay, it's divided into 6 major groups based on their action. Okay, nomenclature based on their mode of action, we can say. The first one is the oxidoreductase. Oxidoreductase normally do the redox reactions. Okay, oxidation and reduction reaction. I think this also you can already see in respiration chapter. NAD, dehydrogenase, or oxidase, peroxidase, those are involved in these groups. Second is a transferase. These transferase transfer of functional group. Okay, they transfer the functional group one molecule to other molecule. Think about kinase. Kinase, what they will do? They will transfer the phosphate group, right? And transaminase, they will transfer the amine group. Okay, phosphorus transfer the phosphate group, like that. Okay. Third one is the hydrolysis. So these normally break the bonds. Okay, break the bond by adding water or by utilizing the water. And that's these are also about, okay, or either add them or remove things, but without water. This question is already asked. Okay, what they are asked, like which particular group of enzymes break the bonds between the substrate without utilizing the water. If it's utilizing the water, it's hydrolysis. If it is without utilizing the water, that's lysis. Okay, lysis means breaking. Next, isomerases, they will convert one form to the other form or change in the isomers. Think about glycolysis. Their glucose is converted into fructose, right? Like glucose into fructose. That kind of enzymes is called isomerase. Last one is the ligases. Okay, ligases form the bond. These are helping the bond formation. I think you already know about in the DNA, right? DNA ligase it and come in the last and it will make a phosphodiester bond. So these are the 6 major types of the enzymes. Okay, then we'll directly go for like how enzymes actually work. Okay, you just have to remember that enzyme never initiate the reaction. Enzymes never initiate the reaction. Initiate reaction. That means what? The reaction should be there. Okay, the reaction should be there. What the enzymes will do? Enzymes increase the enzymes catalyze the reaction, right? Catalyze the reaction. That means they increase the rate of reaction, but they never initiate the reaction. I think everyone of you know about what is catalysis, right? What they will do, we will use in chemistry also we will use different catalysts, right? In the chemistry reaction. Same here. Enzymes are called biocatalysts. They increase the speed of the reaction, but the reaction should be there. They can't create the reaction. Okay. So what here there's a graph. It will show how the enzymes actually work. Okay. Think about this a substrate which has a certain potential energy, and it converts into product with a certain potential energy. Okay, how the conversion will happen? Naturally, this conversion happens when it reaches a particular activation energy, when it receives a particular activation energy and it converts into product. Okay, but when you add enzymes, the activation energy needed to change this substrate into product will decreases. Okay, so that's how with the less energy, what happens? The reaction will happen. To add this point, enzyme never work on. Enzyme never changes. Enzyme never changes potential energy. You just have to remember potential energy is ability of that particular substrate and the product. So it cannot be controlled or changed by the enzymes. Then what enzymes will do? Enzymes reduce the activation energy. They will reduce activation energy which needed to break the bond or to create a bond. Activation energy. They just reduce the activation energy. They don't do anything to the potential energy. Remember, questions are asked based on the potential energy and also activation energy of that particular thing. Okay, so when I consider any enzymes convert substrate into product, substrate into product in a certain rate, right? In a certain rate. So the number of substrate molecule converted into product in a particular time is called as turnover number. Turnover number means what? How many number of the substrates are converted into product by the enzyme? Converted into product by enzyme is called turnover number. Okay. How the enzymes works then? Enzyme work is given by several theories, but the theory which is given to you is lock and key theory. Okay, the theory which is given to you is lock and key theory. You just have to remember this is given by Emily Fisher. Okay, this particular theory is given by the Emily Fisher. So what we will see is like every particular substrate and every particular enzymes have a particular geometric shape, and these act like lock and key. Okay, lock and key. What happens if you put any key, the lock will open? No, right? A specific key is needed to a specific lock. They will also say the same things. For every substrate, there's a particular enzymes will be there, and when they combine, they will form an enzyme substrate complex. This enzyme substrate complex, in that enzyme rotate or change this substrate in such a way that it will initiate the reaction with less potential energy, and after that the enzymes get separated. Okay, you have to remember that enzyme never involved or enzyme never act as a substrate in the reaction. enzyme only facilitate the reaction. Okay, facilitate the reaction. What's the lock and key theory? Lock and key theory says every substrate, enzyme and substrate have a particular geometric shapes, and every substrate have their own enzymes which can fit in itself. Okay, so when we are thinking about factors which are affecting the enzyme activity, there are four major factors which normally affect the enzyme activity. The first one is the pH. Every enzyme works in its own particular pH that we call it as the optimum pH, or you can also call it as the optima. Okay, you can also call it as the optima. Okay, this only in this particular pH only the enzyme work. Think about amylase. Amylase enzyme which is present in your mouth. It can only work in the pH of 7 in the neutral. If it goes beyond that, its activity will decrease, as you can see here. If it is to go below that also, the activity increase. Only in the optimum pH it works or functions in the highest order. The second one which is affected is the temperature. Okay, temperature. Same, every enzymes work in its own temperature. This is called optimum temperature. Again, you can call it as the optimum, optima, or optimum temperature. Okay, every enzyme have its own temperature. Our body enzymes have an optimum temperature of 35 to 37 degrees Celsius. But think about Taq polymerase. Taq polymerase have a optimum temperature of 70 degrees Celsius, right? Taq polymerase, what we used in the PCR reaction, have an optimum temperature of 72 degrees Celsius, right? So it varies by enzyme to enzyme. Above that and below that, again, the activity decreases. Third one, substrate. Okay, third one, substrate. As you increase the number of substrate, the enzyme reaction also increases. Enzyme reaction also increases until a point. Think about like you have 100 people. We kept 100 people somewhere. Okay, and what happens is like you start giving seeds. Okay, every 100 people you are giving 100 seeds, and they are eating it. Okay, think about they are eating it. What happens once you give the 100? Everyone eat the 100. If you give 100, they again eat the 100. Think about you give the 1,000 per minute. If they eat only 100 seeds, after you given thousands also, can they eat 1,000? No, they can only eat 100 per minute because only 100 people are there, right? Exactly same. If the 100 enzyme molecule is there, they can split. They can create 100 reaction at a time only. So as the substrate increases, the enzyme action increases until they become fulfilled. Once they become fulfilled, the enzyme reaction will come to a saturation. Okay, so substrate concentration increases, the rate of reaction increases until the substrate level is equals to the number of enzymes. The fourth one major which is affected is the enzyme concentration. Okay, enzyme concentration. It will show a linear growth curve. Okay, as you can see here, as you increase the enzymes, think about substrate is unlimited. Okay, substrate is unlimited. So if you increase the number of enzymes, the enzyme reaction also increases. Enzyme reaction also increases, or rate of reaction also increases indefinitely. Okay. Is there anything majorly you have to know about it? You have to know about one more thing called Michaelian-Menten constant. Normally, they won't ask about the reactions. You just have to remember, we can discover like there are Michaelis and Menten, the scientists, two scientists given a particular formula for velocity of rate of reaction. V is equal to Vmax of the substance divided by Km plus again substrate. Okay, so what is this Km? Km is called M&M constant or Michaelis-Menten constant. How to find out this? Km equals to Vmax by 2. Vmax by 2. Okay, so as you can see here, this is the Vmax. That means highest rate of highest rate of a substrate enzyme reaction. If you take half of that Vmax, you can find the Km. The Km utilized in a different fashion that we will see in the second one in the enzyme inhibitions. Okay, Enzyme inhibitors are the what? Enzyme inhibitors are the one which hinder the enzyme reactions. Enzyme inhibitors are divided to two types: competitive and incompetitive inhibitors. Competitive inhibitors are the one, these competitive inhibitors replace or attach to the enzymes in their substrate binding site. They block the substrate binding site. So what happens? Now substrate cannot bind to the enzyme. Non-competitive inhibitors are the one which bind to the enzyme other than the substrate binding site. If I show you graph, if it is a regular graph and if it's a regular Vmax showing, okay, if you act the competitive inhibitor, what happens? The rate of the reaction decreases. But if you increase the substrate, what happens? If you increase the substrate, then the rate of reaction again increases. So Vmax will be same. Vmax will be same for the reaction which having the competitive inhibitor or to the regular interaction. Okay. But if you add the non-competitive inhibitor, what happens? It will bind other than the this one substrate binding site. So what happens? The rate of reaction or Vmax decreases. Vmax, what happens? Decreases for the non-competitive inhibitor. Km or Vmax by 2 will be same. Okay, it has the same Km, but the issue what happens is like they have a different Vmax. Okay, so there are some questions are there. I will leave it for the homework because already it's time. Okay, 11 o'clock. I will keep it for the homework. If you have any doubt, you can ask me. Okay, so thank you for joining. Have any doubt, please put that in the chat box or in the WhatsApp or the Telegram. Okay, thank you guys. Thank you for joining. Bye bye.